groups of a human pathogen: enteroviruses and rhinoviruses. Entero and rhino. They are small non-enveloped, icosahedral, and the genome is single-stranded RNA of positive sense. Why? This is a very important point. It is infectious when purified. When we see one of the groups of the family, for example, for poliovirus that cause poliomyelitis, ecovirus, these viruses, the RNA of it is infectious. Even if only RNA infects the patient, it will cause infection. Why? Because the RNA will directly act as messenger RNA because it is positive sense. So does not need a DNA RNA polymerase, only it acts directly as messenger RNA. Proteins, four major proteins, which are VP1 to VP4. Properties of this virus, as we have seen previously, four main protein, VP for proteins, three outside and one inner protein in the capsid. Picornavirus, they replicate in the cytoplasm. They are not inactivated by lipid solvents such as ether because they are highly resistant viruses. They are non-enveloped. Includes three medically important viruses or important genera, as we will take today: enteroviruses, rhinoviruses, and hepatovirus, which causes hepatitis A. We will take it in the next lecture of hepatitis viruses. Hepatitis A is included in the Picornavirus family. If we compare between enterovirus genera and rhinovirus genera, the major groups of enterovirus genera: poliovirus types 1 to 3, Coxsackie viruses A and B, ecoviruses, while rhinovirus contain 100 serotypes. Major group rhinovirus contain 100 serotype, and entero contain three main viruses: polio, Coxsackie, and eco. They infect the enteric tract, found in the nose and the throat. So enteroviruses only in the GIT, the mode of transmission. This rhinoviruses in the nose and upper respiratory tract. Replicate at 37, while these rhinoviruses replicate at 33. Enteroviruses is stable at acidic pH, so they infect in GIT. So survive gastric acid, while rhinovirus is acid labile. These are the main two differences, that the low temperature of replication and they are acid labile. This is the replication cycle. ما مطلوب منكم هذا الرسم بس لنطلع. Picornavirus replication. We'll briefly summarize the replication cycle. First, the replication cycle occurs in the cytoplasm. The virion attach to specific receptor in the plasma membrane. The virion attached to the specific receptor in the plasma membrane. The receptors for poliovirus and human rhinovirus are members of, they are immunological receptors present in the human body, normal receptors. These viruses occupy these receptors. They are member of immunoglobulin gene superfamily, while ecovirus recognize member of integrin adhesion superfamily. Receptor binding triggers conformational change in the cytoplasm, results in the release of the viral RNA, conformational change in the virion, results in the release of the viral RNA into the cell cytoplasm. The infecting viral RNA is translated into a polyprotein that contains both coat and essential replication proteins. Synthesis of a new RNA virus. The virus also forms RNA-dependent RNA polymerase to form a new progeny of the viruses. Then many plus strands get packaged into the virion. The mature virus particles released by host cell disintegrate, that's to say, lysis of the host cell. Usually we see in non-enveloped viruses in general, the damage effect is more severe to the cell because at the end of the application of all non-enveloped virus, there will be lysis of the host cell, lysis of the host cell, which is more dangerous to the human body, more dangerous to the human body. And as we see in poliovirus infection, poliomyelitis. Picornaviruses is divided into two main subgroups اللي هي genera, enteroviruses as we said and rhinoviruses, which we'll all talk about them today. Enteroviruses will include polioviruses types 1 to 3, Coxsackie viruses A and B, and ecoviruses, wide range of viruses. The most important of them is poliovirus. Poliomyelitis, it is acute infectious disease. In its serious form, affects the central nervous system, motor neurons, and the spinal cord, causes flaccid paralysis, but fortunately, most infections are subclinical. There are three antigenic types, as we see it, 1, 2, and 3. Because there is little cross-reaction between the antibody formed against these antigenic types, the three, protection from disease requires the presence of antibody against each of the three types. So, as we will see, the poliovirus vaccine, or OPV, oral polio vaccine, is trivalent vaccine, contain the viruses for the three group 1, 2, and 3. Three serotypes. This picture of a child with poliomyelitis. Poliomyelitis is called in Arabic شلل الأطفال. Pathogenesis. The entry of the virus is through the mouth and replication occur in the oropharynx and intestine. The virus regularly, as you see it, present in the throat and in the stool before the onset of illness. This is very important. Why here question mark? Because what is the danger of this? That means the patient, is still infectious as long as the incubation period and after the appearance of illness. 1 week after infection, the virus continues to be excreted in the stool for several weeks, even though high antibody levels are present in the blood. But the virus excreted in the stool and patient still infectious. The CNS may be invaded by two ways, either by circulating blood to the CNS or along the neuron fibers. The safe virus can either circulating blood or spread along the axons of peripheral nerve to the CNS. So two main ways, where it continues to progress along the fibers of the lower motor neurons. Replication of the virus and the fibers of lower motor neurons to increasingly involve the spinal cord or the brain, mainly in the spinal cord, lower motor neuron. As we see in the motor neuron, there will be damage. The virus invades certain types of nerve cells, they are motor cells, and in the process of intracellular replication may damage or completely destroy these cells. As we said, at the end of the replication cycle of the picornaviruses, all of them, there will be lysis of the cells. Lysis of the cells. As you see, the receptor of the virus on the motor neuron, the virus will attach to them and enter inside the cell and causes cell lysis. Causes cell lysis. طلاب بس سجلوا حضور، انتبهوا قائمة الحضور انقضت قبل 10 من 8:03 يعني فتحت قائمة الحضور. Then, clinically, when an individual susceptible to infection is exposed to the virus, the response ranges from unapparent infection without symptoms to mild febrile illness and severe and permanent paralysis. Mostly, the majority are inapparent. most infections are subclinical. Only 1%, fortunately, only 1% of infection results in clinical illness. So, disease occurs in only 1% of infected subjects. The incubation period, 1 week to 2 weeks, 1 to 2 weeks. Four main presentation. This is very important. In poliovirus, the main presentation are either only mild disease, most commonly form of the disease. The patient has only a minor illness, characterized by fever, malaise, drowsiness, headache, nausea, vomiting, and recovery occurs in a few days. Only mild disease. This is the most common form, which is fever or febrile illness. The non-paralytic poliomyelitis also is common, which is called aseptic meningitis. So polio included in the list of in the differential diagnosis of viral aseptic meningitis. In addition to the symptoms and signs listed in 1, the disease lasts 2 to 10 days, recovery rapid and complete. So it is benign aseptic meningitis. So most common, mild febrile illness. The second, non-paralytic polio, which is aseptic meningitis, mild aseptic meningitis, fever, neck stiffness, meningism sign. The third, which is the least common, only in 1%, as you said, only in 1%, there will be paralytic poliomyelitis, paralysis. The predominating complaint is flaccid paralysis. As you see, there are two main types of paralysis, spastic and flaccid. Flaccid due to damage of the lower motor neurons. Maximum recovery could occur also within 6 months. If there is no recovery within 6 months, there will be permanent damage and there will be residual paralysis lasting longer. So the least common and most important and most severe paralytic poliomyelitis. Paralytic poliomyelitis. As you said, mild, first mild febrile illness, second aseptic meningitis, and then third paralytic poliomyelitis. Fourth, progressive post-poliomyelitis muscle atrophy. As you see, and there is no movement in the muscles, there will be muscle atrophy in the muscles after paralysis. As you see, this child is not emaciated, he is not thin, but there will be muscle atrophy due to low absence of movement of the muscles. Diagnosis, it's not very important, maybe on clinical ground. We can identify the virus in a stool by reverse transcriptase PCR because it is RNA virus. Can be recovered in throat swab also. Isolation of the virus identified by and typed by neutralization and specific antiserum, also by antibody ELISA test. Global eradication is very important. While poliovirus may spread into polio-free countries, so if vaccination programs have been discontinuous, there is a global eradication program supposed to happen at the millennium بال2000 كان المفروض يصير global eradication of the virus, but it doesn't happen. There are still some countries have poliovirus, including also Iraq, still not completely eradicated. هاي أسئلة بعدين نتناقش بيها نحكي properties of polio, virus damage motor neuron because it is non-enveloped virus and causes lysis of the cell. It is what's the mode of transmission of poliovirus? Fecal-oral. Prevention of poliovirus. Both live and killed virus vaccine are present. The most important is live virus vaccine, but killed we give it to immunocompromised children, while live, which is most important. The killed vaccine called formalized vaccine هو SALK. SALK هو killed virus vaccine. while trivalent oral vaccine اللي هو live attenuated يسموه OPV. سابين OPV, oral polio vaccine. Why they give it oral? This is very important point. The live vaccine infect, multiply, and thus immunize, and it induces both humoral and cellular mediated immunity. Not only IgM, but it produces secretory IgA antibody in the intestine. This is very important, secretory IgA antibody in the intestine, which then become resistant to reinfection. Why? When the port of entry of the virus by the fecal oral route, so we induce by this vaccine secretory IgA antibody in the gut, in the intestine, so we'll destroy the virus in its site when at the site of entry to avoid spread of the virus to the blood and then to the CNS. At this site, we will destroy the virus by this vaccine, by this formation of secretory IgA antibody. This is oral polio vaccine. طبعا repeated doses are very important. Booster doses is very important. And هاي أشياء بس طلعت تقرون عنها. These are the end of poliovirus that causes poliomyelitis. The second group or in the
دكتورة اسمها العفو.
نعم عيني تفضل. نعم.
الدكتورة الكلب فاكسين يعني إذا انطوا انجكشن
نعم.
وسوى الاي جي جي والاي جي ام،
نعم.
واذا كان النقل مال الفيروس عن طريق النير فايبر، شنو الفائده انه راح نعطي كلب فاكسين؟
لا هو could be ممكن نعطي injection killed virus vaccine formation of IgG and IgM antibodies, so they will kill the virus also. Therefore, but if we give it orally, we prevent the virus better. Okay. عندك يعني أنا ما فهمت سؤالك ممكن بالله بس
دكتورة يعني قصدي احنا قلنا عندنا تو واي اوف ترانسميشن تو ذا سي ان اس، يا اما عن طريق البلاد يا اما عن طريق النير فايبر قبل من الجي اي تي.
نعم.
يعني اذا صار بالترانسبورت عن طريق النير فايبر فهو ما راح يوصل للبلاد بهاي الحالة.
لا هو should be absorbed from the GIT into the blood, then it will be transmitted from the blood to the nerve fibers. either يعني أكثر شيء هو أهم شيء من البلد to the nerve fibers، وبعدين يقول لك هو to the CNS، يعني إذا أحكي هو transmission to the CNS either directly from blood or from the nerve fibers. So it should be absorbed from the gut. فإحنا نمنع يعني نقتل الفيروس بالفاكسينيشن بالاي جي antibodies directly in the gut. فهمتني؟ يعني هو
تمام دكتور، يعني لازم يمر للبلاد.
طبعا لازم يمر للبلاد، بس من البلاد two ways, either to the CNS passes the blood-brain barrier أو to the nerve fiber, peripheral nerve fibers، وبالطريقتين هو راح يوصل to the CNS. أوكي. وهو طبعا التارجت مالته هو التارجت هي الموتور نيورون posterior motor neurons that may causes severe paralysis. أوكي. أكو سؤال أيضا؟ تفضل. أكو سؤال بعد عن poliovirus. Now we'll talk the second group in enteroviruses, the Coxsackie viruses. They are a large group of viruses that contain two main types, A and B. They can be also transmitted by fecal oral route and could be by respiratory aerosol. Pathogenesis, the virus has been recovered from the blood at the early stages of natural infection. Virus also found in the throat for a few days early after infection and in the stool. The virus distribution similar to other enteroviruses in the whole body could be. So, a picture of Coxsackie viruses in the stool. Let's ناخذ محاضرة electron microscope EM picture and we'll talk about them clinically. First, a group A specific disease اللي هي group A اللي group A Coxsackie viruses. we will بس احفظوا حرف ال H. Group A H, Herpangina and hand, foot, mouth disease. Herpangina and foot, mouth disease. Herpangina includes fever, sore throat, and tender vesicles in the oropharynx. While hand, foot, mouth disease, it is vesicular rash on the mouth, hands and feet, ulceration of the mouth. Herpangina هي mild form of hand, foot, mouth disease. Herpangina, fever, sore throat, and tender vesicle in the oropharynx. While hand, foot, mouth disease, more severe vesicular rash Severe vesicles occur on the hand and feet and the mouth with ulceration. This is hand-foot-mouth disease. The Group B, Coxsackie viruses. Group B mainly in the heart and lung. In the lung causes pleurodynia, or what is called pleurisy, fever with severe pleuritic chest pain. Clinically, what is pleuritic chest pain? It is pain on deep inspiration. Pain, it is very characteristic, pain on deep inspiration. If there is cough هيصير أيضا during the cough there will be chest pain. The second, myocarditis. All these are group B Coxsackie viruses. Pleurodynia, myocarditis. Myocarditis, inflammation of the heart muscle, and also could be pericarditis. But all of them are mild self-limited diseases. But the permanent damage caused by Coxsackie B is type 1 diabetes. Diabetes ومرض السكر, diabetes mellitus. The virus suspected to have a role in juvenile diabetes. واحدة من أسباب juvenile diabetes type 1 diabetes mellitus هذا طبعا proved هو Coxsackie viruses type 2 type B. Disease caused by both groups A and B could be aseptic meningitis, as we said in the previous lecture. يوم الخميس تكلمنا عن الاسeptic meningitis وقلنا واحد من ال differential diagnosis aseptic meningitis are Coxsackie viruses, both A and B. And they both could cause mild upper respiratory tract infection with or without rash, with rash in children mainly. These are main diseases caused by Coxsackie viruses. نحفظها type A حرف ال H. Okay, where type B in the heart and lung and diabetes. Lab diagnosis. Rise in antibody titer. Further be by ELISA and could be by PCR to detect the virus. Treatment, there is no antiviral therapy or even a vaccine. طبعا both of them, poliovirus and Coxsackie virus, there is no antiviral vaccine. No antiviral therapy. Polio, there is vaccine, but Coxsackie virus, there is no vaccine. سؤال عن Coxsackie viruses فقط هذا المطلوب من عندكم عن الكوكساكي viruses. The third one in the enterovirus family is the ecoviruses. Disease viruses cause variety of diseases, such as mainly aseptic meningitis, and could also cause infantile diarrhea. Aseptic meningitis, upper respiratory tract infection, febrile illness with or without rash, and infantile diarrhea. And it may cause rarely hemorrhagic conjunctivitis. منو عندنا الفيروس اللي ذكرناه بالسابق causes conjunctivitis؟ حكينا عنه الادينوفيروس.
الادينوفيروس.
نعم شكرا. والإيكوفيروس the second one, it causes also conjunctivitis. Ecoviruses طبعا هي wide range of group, mainly transmitted by fecal oral route. They are one of the leading causes of aseptic viral meningitis. This is most important disease caused by ecoviruses. طبعا receptor of ecovirus هو integrin adhesion superfamily وهو receptor of the immunological receptor. Now, the second genera in the Picornaviruses. As we talked all of these before, the poliovirus, Coxsackie viruses, and ecoviruses all are in the genera of enteroviruses in Picornaviridae family. The second one genera in the Picornavirus family are rhinoviruses. As we said previously, when comparing between rhinoviruses and enteroviruses, rhino more than 100 serotypes, different serotypes, and they replicate in the nose and throat. They are acid labile, so cannot be infected through the GIT, only transmission through the respiratory tract. And finally, most importantly, the replication temperature. Rhinoviruses rhinoviruses, replicate at 33 degrees centigrade, while enteroviruses can replicate at 37 degrees centigrade. Rhinovirus group disease main cause of common cold. يحصل عندنا only running nose and rhinorrhea and could be sneezing and coughing. Properties of these viruses, more than 100 serotypes, replicate better at 33 than 37, and they are acid labile. They are non-enveloped RNA viruses of positive sense single-stranded RNA. Non-enveloped RNA, single-stranded positive sense, very important. The same properties as virological properties as in enteroviruses, the same properties of the genome. Replication, the cell surface receptor is ICAM1, intracellular adhesion molecule 1. أيضا it is immunological receptor. As you said previously, الإيكوفيروسز integrin adhesion molecule وهذا الرينوفيروس receptor is the ICAM1 receptor. ICAM1. Transmission, two modes of transmission. Direct from person to person by sneezing, coughing, respiratory droplets through aerosols. And could be also indirect. Why? هذا more common. The respiratory droplets are deposited on the hands and surface, then transported via fingers, nose, and eyes also, and transmitted to the human body, either directly from person to person or indirectly by the virus present on the personal equipment of the infected person and could be transmitted to other persons. The virus is non-enveloped, so it is resistant and could be living outside the human body for very few hours, so that could be transmitted through the equipment rather than from person to person, more commonly. Clinically, after incubation period of very short, 2 to 4 days, there will be sneezing, nasal discharge, sore throat, and could be cough. Cough is mild. Headache is also common. So there will be sneezing, nasal discharge, mainly. The illness lasts about 1 week and it will be mild, self-limited. There is no antiviral. There is no vaccine. The vaccine is difficult for this virus because it's very wide range of types of virus, more than 100 serotypes. So it is very difficult to give vaccine for rhinoviruses. طلاب بس وقفوا التسجيل يعني حتى ما تصير طويلة عليكم we'll start عندكم أسئلة عن الفيروس عندكم أسئلة عنه أكو سؤال سجلوا حضور
بس قلت جينوم مالته شنو؟
نعم الجينوم is single stranded positive sense RNA virus single stranded positive sense RNA viruses كليتهم هذول طبعا وهم كلها non enveloped فتكون resistant viruses دائما two main important properties بالفيروسز اللي هي non enveloped أول شيء تكون resistant to heat resistant to mainly gastric acid except rhinoviruses so and it could live on the surfaces outside the human body extracellularly for short period of time so transmitted by this way and this is the first point تخليها دائما ببالك من تعرف بأنه it is non-enveloped virus لكن the second point if it is non-enveloped virus that the virus causes damage, severe damage to the cell infected by the virus. That because at the end of the replication cycle of the virus there will be lysis of the cell. وهذا طبعا most important point لأن poliomyelitis مثلا بالبوليو ليش يصير paralysis ولو هي rare only in 1% of infected person but it causes lysis of the motor neuron and damage of the motor neuron and it will be permanent damage. طبعا نجي شوي نتحدث عندكم سؤال بعد نحكي شوي عن ال OPV oral polio vaccine. Oral polio vaccine, very important point about oral polio vaccine is that it is live attenuated vaccine trivalent for 3. من نسمع live attenuated معناته راح يحفز لنا الcell mediated immunity. لكن دائما ننتبه بأنه نقطة جدا مهمة حفظ the preservation of the vaccine when it is given to the children. لازم يكون at small refrigerators اللي هي transportation refrigerators, very important because damage of the virus is very common, of the vaccine, damage of the vaccine is very common. The second point in the vaccination, the child, if he is vaccinated, it will be source of transmission of the virus, especially for immunocompromised and elderly persons. أيضا there is a risk in elderly person more than 70 and 80 years. دول risk إذا هم مثلا ويا الأطفال الصغار أحفادهم وياهم عايشين بالبيت, they are vaccinated, هذول could be risk of transmission of the virus to elderly, and though it is very rare, but it could cause paralysis in the elderly persons, the vaccine itself. And also it is more dangerous for immunocompromised, واحد on immunosuppression therapy. مثلا احنا عندنا أكثر شيء عندنا kidney transplanted persons or person on chemotherapy malignancy on chemo. لا ماكو عرض, انتهى العرض, بس نتحدث عباس, نتحدث بس يجي كأسئلة ومناقشة خصوصا على البوليو فاكسين. اكو سؤال طلاب ايضا بعد سؤال ان شاء الله ما اكون يعني ما مرت استعجل بالمحاضره, بس قلت حتى يكون التسجيل قصير وينزل. انا نزلت لكم الباوربوينت الباوربوينت حنزله عندكم, وتسجيل بعد انتوا عندكم التسجيل راح يصير. اوكي اكو سؤال؟
تمام دكتورة, شكرا, واضحة.
شكرا جزيلا, يعني مع السلامه.
الله وياك.